My name's Tyrone DeSpenza. I'm a MD PhD student at Yale. So that just means I did the first 3 years of med school, and I started my PhD in the neuroscience program in January. And my mentor, research mentor, is under the guidance of Kristopher Kahl. He's a pediatric neurosurgeon at Yale. And basically, in the Kahl lab, we study the genetic basis of neurosurgical disorders that are pediatric. And so I'm going to talk today about the genetics of Chiari Malformation. And so my objectives are to provide some evidence to show you that Chiari is a genetic disorder and that it's largely undefined. And in hopes of my thesis project is we hope that identifying genes will help us better understand Chiari so that we may be able to improve diagnosis and advance treatment options in the future. And in my talk, I will also provide you with previous Chiari genetic findings and also tell you about what we're doing in the Kahl laboratory at Yale. So this slide just talks about basically to show you some evidence that Chiari may be genetic in part. It's a Mendelian disorder, which is inheritable, like a disorder you inherit, which is caused by a variation within a gene. And Chiari is associated with many other Mendelian disorders, such as Robin sequence, NF1, Noonan syndrome, Ehlers-Danlos. And these genes in red, these are genes that are associated with these disorders. And largely these disorders are involved in craniofacial abnormalities. And there's been multiple case reports of Chiari diagnosed in identical twins, and also a case involved in identical triplets. So one study in 2011 showed that out of 500 pediatric patients with Chiari, the prevalence of familial Chiari, meaning more than one person in the family had Chiari malformation, was about 3%. And so there's this interesting connection between Chiari malformation and connective tissue disease. So in a previous study in 2007, they showed out of 2,813 patients, 5% of them had Chiari malformation and some type of hereditary connective tissue disorder. And of them, Ehlers-Danlos was the most common, and that's a connective tissue disease that causes spectrum disorders due to mutations in collagen genes, and it's thought to cause Chiari through cervical instability. And to date, actually the largest family reported in literature had 5 individuals affected with Chiari, and there was an extensive family history of Ehlers-Danlos. So this is basically a family tree of that study, and you can see that the dark circles and squares, those are individuals affected with Chiari malformation. And so before I get into the genetics of previous studies, I want to review some terms. So there's this thing called a single nucleotide polymorphism, which is the most common genetic variation you can get, and people call these things SNPs. And so it's a variation in a single base pair or a building block of DNA that may or may not be associated with a disorder. So in this example, this cartoon, I showed 3 people, and you can see here there's an A, G, and T. They're different, and this is what we call a SNP. This variation here, A, G, and T. And in previous genetic studies, you could study these SNPs using genome-wide association studies, or GWAS. And GWAS used dense array of common genetic markers, or SNPs, to identify locations on chromosomes which are a part of DNA that are associated with a particular disease. So if one type of variant is more frequent in people with the disease than people without the disease, which is the control group, the variant is said to be associated with the disease, but this does not mean causality. So this, just because there's people with SNPs in the gene doesn't mean that gene is actually responsible for their disorder. And I found this on the ASAP website, actually. This quote is, "A GWAS study is akin to surveying one household in every block across the United States. An initial analysis will point researchers to the right neighborhood where they can go and survey each house on the street." And right here, I show a Manhattan plot. So basically, these are all SNPs. And the ones here are the ones that are found more frequently in basically in people with a particular disorder. And so there's been a few previous GWAS studies done in individuals diagnosed with Chiari malformation. In summary, a lot of these genes that were discovered were in collagen connective tissue. So FBN1 is a known gene in Marfan syndrome, huge connective tissue disorder. There's also GDF3 and GDF6 also involved with the connective tissue and found in Klippel-Feil syndrome, which is on a spine disorder of the bone. And there's these other genes too involved with basically collagen, and these genes are involved with development while you're while in utero or while you're still in the womb. And it's pretty interesting that these genes have been associated with Chiari so far, but it hasn't been proved that these genes actually have caused Chiari. And the reason with that is because in GWAS studies, you find a lot of the common SNPs that are found in a lot of people that usually aren't causing disease. So in GWAS studies, it basically in this diagram I show, adapted from McCarthy et al., that the penetrance or what the disease you see on the y-axis, and on the x-axis I have the allele frequency, basically how rare or how common these genes are. In a GWAS study, what they will do is you will be able to identify a lot of the really common variants, but these variants only have a very low to modest effect on the disorder. So they don't really explain the disorder in its entirety. So what you want to do in a Mendelian disease is basically you want to find a very rare variation in a gene that may be able to explain it. And so that's what our lab does. We focus more on the very rare genes that are found in people with Chiari and try to identify which genes or pathways that have variations in them, so that we could then target them and better understand it. So there's been groups around the world who are using this whole exome sequencing approach as well. And this group is from Italy. What they do is they have these two families again. So these dark boxes means these individuals in this family have Chiari malformation. And what they've done is they've done this thing called whole exome sequencing. And so what whole exome sequencing does is you look at the region of DNA that actually is expressed, so meaning the DNA that actually becomes genes. And in those genes, 85% of mutations in those genes actually have been associated with different disorders. And basically, in this study, they found that there's genes related to that are really responsible for craniofacial development, which is interesting. So with that in mind, at Yale, for my thesis project, we used a very similar approach in the whole exome sequencing. It's NIH-sponsored at the Yale Center for Genome Analysis, and it was established about 6 years ago, and as of January 2017, at 5 years after its inception, there's been 91 projects, 92 collaborating investigators, 8 countries, 69 institutions, 75 publications then. Now it's well over 200 publications. And more impressively, 461 novel genes have been discovered. So this was started by Richard Lipton and Shrikant Mane at Yale. And basically, in collaborating with the Yale Center for Genome Analysis, or YCGA, we are able to do our Chiari genetic study as well. So I started my PhD in January, and since then, I've been able to meet 24 families with Chiari malformation. And a familial Chiari is basically you have more than one individual in the family with Chiari malformation. So in a very short time, within 6 months, I've met all these families, and these, like before, so these drawings are just family trees, if you will, or pedigrees of the Chiari malformation families. And the dark boxes, again, are people who are affected with Chiari malformation. And the white ones are the people in the family that don't have it. And so we get DNA from these different individuals, and we can also see, in mapping out their family trees, we can see how the Chiari is inherited in those families. see here is passed down from the mom to all the children, and even in the grandchild here. And so we like to map this out so we can see the inheritance of the Chiari, and also in doing so, we can basically figure out to see if there's any genes that are common amongst all these dark boxes or people affected with Chiari malformation. And so one of my first, actually my first project in the lab about 2 years ago was we had a patient with Dense disease, and he also had Chiari malformation, and we did perform whole exome sequences analysis at YCGA, and we found basically we got DNA from his dad, his mom, and the child with Chiari, and we were able to find this thing called a de novo mutation. And so what a de novo mutation is, it's a mutation that's very rare. It's like 1 in 100 million. And basically, these are rare spontaneous mutations which could potentially cause disorders. And parent-offspring trios, so having the parents and then having their children affected with Chiari, is a really effective tool to identify genes that contribute to Chiari malformation. And we can do this. It's very effective because what you can do is you can find de novo mutation, let's say in this individual. If another individual has the same de novo mutation, that same gene, the odds are is that it's not due to chance and that those genes are probably causing the Chiari malformation. And so we recently used this approach. The first project in the lab before I started in January was with a medical student, Shrita Fury. She's now a neurosurgical resident at Barrow. And what she did was she basically got 125 trios, so the mom, the dad, and the affected child with Chiari. And she used whole exome sequencing to find de novo mutations in the affected Chiari patient. And in doing so, she was able to identify 4 novel genes that cause 10% of congenital hydrocephalus, and she also identified new pathways that are targetable by FDA-approved drugs for congenital hydrocephalus. And this, her article was featured on Neuron earlier this week. So if you want to check out that article and learn more about our studies, I want to do the same exact thing for Chiari malformation. And so in future directions for my thesis project is we're looking for more Chiari and Syringomyelia patients to join our study. And to join our study, you don't have to live near Yale or actually in the United States. And even if you don't have a family history, we're interested in your participation. So anyone you know with Chiari malformation, or if you have it yourself, it's really free and easy to join our study. All you'd have to do is send me an email at chiari.genetics@yale.edu, and I can send you information about our study and our consent forms. And if you're interested, the next step would be to, we send out these little buccal swabs. And on these swabs, basically, the way it works is you email me, and I email you back with all the information. If you decide that you want to continue with the study, you would give me your address, and I would send a prepaid FedEx packaging to your house that has these buccal swabs. And what we do is you take the buccal swab and you swab each side of your cheek for 30 seconds like so. And it's not painful at all, and it's really easy. So 30 seconds each side. You then place the swab back into the container, pull out the stick, close it, and label it with your name. You put it back into the FedEx packaging that's prepaid, and you sign the consent forms. And from there, we can isolate DNA, do whole exome sequence analysis, and figure out what genes are responsible for the Chiari malformation. So it's really simple to join our study. And currently, like the 24 failings I showed earlier, they were largely in part with collaborators, Dr. Butler at Harvard, Dr. Jackson at Hopkins, and in Wisconsin. And we're really open to collaborating with any physicians here at ASAP as well. So thank you so much to ASAP, the YCGA, Richard Lipton, Shrikant Mane, Hopkins, Wisconsin, Harvard, and our Kahl lab. Thank you so much.